Yeah, thank you. Thanks for inviting me. Can you hear me? Maybe someone can just give me a confirmation. Yes, perfect. Thank you. Hi, so my name is Monika Huhn. I'm a data scientist in the Data Science and AI department at AstraZeneca. And today I'm presenting OneView, which is a Shiny application that we created for drug repositioning investigations. And I'm speaking on behalf of a quite large cross-functional team. So this was a bigger project that involved a lot of people and several outputs actually. Okay, so I have only 10 minutes for this talk and I have a very ambitious number of slides. So I will have to go very quickly and probably won't be able to take any questions, but I'll see what I can do. But happy, of course, to answer any in the chat afterwards. So I'll start by going here. So first of all, a short basics on drug repositioning. For those of you who have never heard of this before, so what is drug repositioning? Very shortly, it is around finding new disease indications for a drug that is already known and approved for something else. We actually call it digital drug repositioning for this project. And what is the digital referring to? So we are looking here at connections between diseases and drugs that are not previously known. And in order to find those, we have to look at many possible connections. And then obviously when you're having to look at a lot of these, you will want to use computational methods to achieve that. And obviously for that you need data. So what kind of data are we talking about when we're talking about drug repositioning? And actually this is a wide field. There's enormous amount of publications with all kinds of data, be it gene expression data, proteomic data, real world clinical data, and many more. So really whatever data is available can probably be used in some way or another. And what's the importance of this, just quickly to give a little bit of a purpose statement here. So really the main purpose is to save time and costs, and the benefit of that obviously is that patients will have a new treatment much faster, and also it enables you to look into treatment for rare diseases. So now specifically to the project that we're working on, OneView. What was this project about? So first of all, we're going to take a little look at the data that we used. So in this specific project, we used transcriptomic data. We had data that was generated for our own compounds by treating cell lines and basically comparing the gene expression levels before and after treatment. And then the same, and then we matched those signatures with signatures from disease samples. We would also compare the gene expression levels of patients with disease versus those that don't have that disease. And when we're looking at a matching, we're looking at an opposite pattern or kind of lock and key pattern match, where you, on this graph here, this would be the left example. So you want to see examples where in one of the two, the gene expression levels are up regulated and the other one are down regulated rather than in the same direction. And what specific problem did we try to solve in this? So the matching was actually not part of this specific part of the project. That had already been done. So OneView came in place after this with the goal of understanding what, when we see mappings, what drives these mappings on a gene level, and then also can we find further evidence for these connections. And how we worked in this project was with the cross-functional team, and this explains a little bit also the problem that we were trying to solve. So we had two types of people involved in this project, with biologists who understood kind of the significance of some of the mappings, but it was a lot of data that they couldn't easily work with manually. And then we had data scientists like me who knew how to handle the data but didn't understand the exact biological questions. And then the whole process of this then was of us working together iteratively using a lot of prototypes and especially visualizations to facilitate communication, but also to find the best solution, and then finding basically a solution that enabled the biologists to do their work. And that solution is OneView, which is a shiny app that allows the scientists to easily access all of the data, filter through it, search through it, follow through different data sets. So really a way for them to work with this abundance of data that has come their way. And we also, on the point of substantiating with further evidence, we also managed to loop in some of our other internal databases, such as our knowledge graph and also our omics signature database for further evidence. I want to briefly also go a little bit more on the technical details on the R side, which is probably most interesting to most people in this audience. So first of all, why did we build a shiny app? As probably most of you know, these are very easy and quick to build. So that was a big win for us, of course, but we also wanted to be able to customize it in the way that we wanted, which also Shiny allows. Some packages we used was the DT package for the data table functionalities, Shiny widgets for some extra. We also wrote our own style sheets, and we also extended functionalities using HTML and JavaScript, and more specifically D3 JavaScript visualizations, which I will show you in a minute. We also have a good infrastructure within AstraZeneca for deploying Shiny apps, so that was also a big, that made it also easier to work in that way. And lastly, we really wanted to use the R functionalities when it comes to data manipulation and data preparation with the tidyverse packages. So yeah, so that was very handy as well. So as I mentioned before, we have focused a lot on visualizations and trying to visualize the data in a meaningful way. And then obviously one of the most natural way of visualizing data that shows connections are network graphs. So this is an example of a network graph from OneView. It is based on simulated data, so don't try to interpret this in any way, but I just want to show you this a little bit. So this is actually a D3 visualization, and as you can see, it's quite handy because you can do some zooming and changing around, but most importantly, it is completely interactive. So if you click on things here, you will get further information very easily and very quickly. And there we go. If I have time, I will talk a little bit more about the D3 connection. And then one other visualization we focused on a lot in this tool is the so-called upset plot, which is a generalization of Venn diagrams for more sets. So you see a Venn diagram here for 5 sets, and it's quite difficult to see which intersection represents which sets. And the upset plot on the left is basically showing the same information, but it's showing in the matrix plot below, it shows which intersection we look at, and then in the bar plot above, it shows what these mean. And in the standalone R package for upset plots, it's not interactive. So that was one of the main reasons that we went for building this in D3 instead, because we wanted it to be interactive. We wanted to be able to click on things and find more information. D3 is very nice because it gives you really beautiful visualizations, and the data is directly bound to the graph, so it's instantly there. But it's not the easiest to work with programmatically. So there are some pros and cons. And I'm almost out of time, so I'm going very quickly here now. So lastly, just some information how we did this. So we did all the data prep in R using the strength of R, and then we sent the finished data over to D3 to display the visualization. For this, we used a session object handler function. There's also an R package called R2D3, which could be used for this, which we haven't yet, but we're hoping to do that in the future for post-processing, sending this back into the Shiny app. Yeah, so I made up good time. So I think I'm exactly at the half an hour now. So thank you, everyone. I hope you can see this nice D3 animation, I think, is lagging a little bit on the connection, but yeah, thanks a lot. And if you have any questions, I'm happy to take them in the chat or in a separate session.